Hey, welcome back, guys. As promised, we are going to do part 2 of this video, and that will be answering your questions from part 1. But of course, we're going to do it in a different way because in part 1, we learned the science and biology of the coronavirus disease 19 and Sars Cov2. So you will be able to answer a lot of these questions based on what we learned in the previous video. So it doesn't have to be anecdotal. It doesn't have to be just somebody you trust. You will know because you can answer it from the actual science. And that's what we're going to try to do in this video with answering some questions. Before we begin, just a very couple of quick updates and also housekeeping. The first one, there was a lot of concern after the posting of the first video when the video was taken down very soon after, and there were all these conspiracy theories of YouTube is taking it down because they don't want accurate information getting out, all kinds of things that were kind of flattering. But unfortunately, as usual, there is a logical scientific explanation. I had noticed I made a rather egregious math error in the epidemiology section. And because I want this to be accurate, and I also don't want people to look at it and then have doubts about it because of a gross inaccuracy, I thought it best to pull the video down, make the correction, and then repost it. So that's what happened there. No conspiracy theory. Even then, there are some small mistakes. I think I said 2018 instead of 1918 twice in the video, even on the correction. But anyway, those are pretty minor and obviously errors, but the math error I made was fairly egregious, so that explains that. The second thing, I want to make it very clear that while I'm sure a lot of you guys are following along with some of this fairly advanced science that we're doing, gene sequencing, gene expression, indels, all of this stuff, keep in mind that I am still very much oversimplifying this. For example, you guys now know how antibodies work through the lock and key method. However, what you aren't aware is that there are lots of types of antibodies, IgGs, IgMs, polyclonals, monoclonals, oh my gosh. We're not going into that. The idea is to get you enough of the science that you can identify misinformation. That is the purpose in this video, to give you just enough that you can say, wait, I know how this works. That is wrong. And maybe help to fight against the misinformation. The misinformation, I believe, has an R naught far greater than the virus does. Again, I did caution in this part of the video at the very beginning in part 1 that I was going to oversimplify, and that if you guys are familiar with genetics, maybe you need to let some of the accuracy slide a little bit. But I had mentioned that the reason we know that the bat coronavirus was the source for Sars Cov2 is because of a 100% homologous sequence that the human virus has with the bat coronavirus. And somebody had pointed out, somebody who actually is familiar with looking up things on GeneBank apparently and has scientific background, that he was only able to find that there is a 96% homology. And by that I mean the sequence, the RNA sequence, the ACGU, GGGC that we covered, that it is actually only 96% homologous with the human version of the coronavirus. And two things about that. First of all, yes, I did kind of exaggerate the accuracy a little bit, but I didn't want to go into variants of there's many different bat coronaviruses. The one that I had actually used is it shows on GeneBank, and I will show you, it actually is 99.98% homologous, the bat coronavirus to the initial human coronavirus in Wuhan. From nearest as possible, I could find to patient 0 that was sequenced. The GeneBank ID, if you want to look, again, everything I'm saying is verifiable and falsifiable, so you can look it up. The GeneBank ID is, let me see here, MN996532.1 that I had used to find the closest homology for that example. Even so, couple things. First of all, 96% homology, even if that's the highest it is, doesn't matter. It still verifies it came from bat coronavirus. That is the point. It is to get rid of the misinformation that this came from other sources. The second thing about it is that the amino acid sequence, remember the lock and key, the conformation of the protein, remember from the first video? The amino acid sequence is what is important, and that is like 99% homologous. The third and final thing about it, I actually forgot. What is it? Oh, yes, I remember now. The third and most important final thing about the sequence homology is that it doesn't matter from the bat. When we're talking about a mutation in the virus, especially a mutation that would cause a novel version of the virus, you don't compare it with the bat. You compare it with patient 0. So in other words, the first transmission to a human, that's your baseline. Any mutations from that one are the ones that you are really worried about, right? And I am going to show you right here conclusive evidence that it is not mutated. Okay, so don't send me any more things about asking about mutation. Check this out. Okay, hopefully this shows up clearly in camera, but you can see here the actual gene sequence from the Wuhan coronavirus, from basically patient 0, the first human transition, the person that had the disease and then the virus was recovered and sequenced. We can see that there are T's instead of U's because you cannot sequence RNA. I'd mentioned that in the previous video. You have to reverse transcribe it to DNA, so all of the U's will be converted to T's. This is the actual sequence. By the way, error I did, again, just because I was going from memory and estimating. We can see, let's page down here, we can see these numbers on the left indicate the position, the number of the base. You guys might remember I said the sequence was approximately 24,000 bases. I actually underestimated that. There's 24,000 right around there. It's actually about 30,000. We see it ends here with the poly A tail, if you're familiar with that, at just about 30,000 bases. And actually, I accidentally lost part of my protein sequence. Again, this here is the codon sequence. I missed some of it here. So if you guys go and try to match it up or something with the DNA, you're welcome to try that. It probably won't work because I missed the first part, I believe. But this is the protein for the virus broken down by the amino acids. These are the sequence of amino acids for every 3 bases, and so on. What I did here, and actually let's go to the actual gene bank in NCBI. What I did here is I ran 2 sequences together. It's called a basic logical alignment. And what I did was this top sequence here, which goes along just the top here, that is the Wuhan sequence. The Wuhan Sars Cov2 recovered from one of the earliest cases of Covid19, and that is the sequence that was derived from the virus that infected this person. The lower strand is the sequence from just somebody that I pulled from the United States of America. I'm not sure how recent or whatever, but obviously different continent, much later than the Wuhan one would have been. So by this time, you've got tens of thousands, if not hundreds of thousands of people infected for sure. The top strand and the bottom strand, again, are 2 different people. If the nucleic acid from the top patient matches the nucleic acid from the bottom patient, it's also indicated by these lines. So if you see any gaps in these lines, then that indicates that there is a difference in those sequences, and it could be a mutation, it could actually even be an error from the sequencing. The sequencing has an error rate of, in some machines, as high as a couple %, actually. So do keep in mind, just because you see any differences, it doesn't mean it's a mutation. It's more likely actually an error. In this one, however, we can see we are going to have no gaps right here. It is 100% homologous. There are no mutations. Even if there was a mutation and it wasn't an error, in sequencing, it was a legitimate mutation, remember, it has to change the amino acids and even then change the conformation of the protein to make it unrecognizable by the antibody. That is not even the case. I'm going to page down, just going to scan along. Don't even have to look because we already know there are no differences between the initial patient 0 and recent case. Now, there are some I've seen where there was 99% homology, so there was a 1% variance in the sequence, but again, no indications that that is beyond the sequencing errors as well as the fact that the amino acid sequence was still conserved or the same. All right, this goes on again all the way to 20. If you see a missing base or one that is mismatched, let me know. Okay, I have shown my evidence that is publicly available. Anybody in the world can access that and they can validate it for themselves. All I am asking is that if you still insist that the virus has mutated into a novel form already, or 3 of them like one person was trying to tell me, I'm just asking you to give me the evidence because we never see ever evidence of this. I'm a big boy. Give me the gene bank accession number. I can find it on my own. Nobody ever does that. It is strictly anecdotal. It is unscientific. So with that in mind, we are going to now bring in our special guest. And what we are going to do is ask some questions from the previous video. But because this special person, who is not a scientist, watched the video, based on the education she now has, we're going to see how many she can answer and if she gets them right, because you guys should be able to do the same. Let's do it. Okay, guys, we have our special guest. I think a lot of you guys knew this was coming. This is my girlfriend, Vicki.
Hey, guys.
Many of you subscribers probably remember her from a previous video or two. So, Vicki, thanks for doing this. The purpose here is, Vicki, you're not a scientist, right? Just checking.
Absolutely not.
Okay, so Vicki is a lay person like many of you, but she did watch the video. After watching the video, her confidence in being able to identify a lot of misinformation and question things, she says, was dramatically improved. So we're going to some degree test that and actually have her answer your questions with a little supervision and see how well she does. So some of them she won't be able to answer, though, because they weren't covered in the video. So those I'll take. As a matter of fact, let's go ahead and get started. She made the question list. I don't know what she's asking. She just thought they were interesting.
Okay.
All right.
So the first one, is PPE, personal protective equipment, effective?
Is PPE effective? Okay, so here is a great start for this video because, again, I am going to give you real, accurate information, but you sometimes have to be responsible for it and also take it just at the scientific level. I'm well aware that there is a lot of information out there from experts that wearing PPE has no effect, and I am going to tell you that they are actually right, but for the wrong reason that you're thinking. PPE equipment is protective. It does help. Wearing a face mask, wearing a glove, absolutely helps you to not get infected from the virus, period. End of story. I honestly don't care what the experts say about ineffectiveness of masks. I have worn masks almost every day of my career, and I know what they do. I know how they work, particularly the N95 masks. However, that said, for most people, overwhelmingly, they're not only going to be ineffective, they're actually going to be counterproductive and could increase your risk. So first of all, why would they be ineffective? It's because people don't use them right. People don't use the PPE right. So as an experiment today, Vicki and I went shopping. We went to Costco, and we wanted to use proper, what is called sterile technique. We want to make absolutely sure we do everything to minimize potential contamination. And we're seeing all of these people in the store that have their masks, their gloves, even protective eyewear. First of all, Vicki, you want to talk a little bit about the experience of shopping with correct technique and how that was, and then learning that technique, what you noticed about other people. By the way, I don't have to mind the social distancing here, and it's very hard to.
So shopping with
proper technique was interesting because you don't notice how many times you touch your face, you touch items, you touch doors, you touch tables, you touch the cart, then you touch your face again. You don't notice that you're doing that. Mm-hmm. You take out your hand sanitizer and you do it again, and you think, I'm not going to do that again. But you do. And everybody else already has. So it really doesn't matter when you're in that kind of environment. You're not in a controlled environment, so no matter what you do, it's not going to matter.
Pretty much that's true. And not just that, but let me expand on this a little bit. The improper use of the PPE is what the problem is here. That kind of defeats the purpose of even using proper technique within a grocery store. Remember the lady that had the N95 face mask, the gloves, the eye protection, and then her cell phone rang. She was picking up all these items in sterile technique. When you put your gloves on, as soon as you touch something, anything that isn't sterile, the assumption is you just loaded your gloves up with coronavirus. And what did she do? She fumbles through her purse, contaminating everything in her purse, gets her cell phone, contaminating her cell phone with her dirty gloves, pulls her mask down, talks in the contaminated phone with her contaminated hands that were touching all these products, and then puts the phone back, takes her fingers, pinches the mask to pull it back up over, trapping any possible coronavirus inside her mask so that she can only potentially trap it and breathe it in. And we saw this
multiple times.
All over the place. Also, the checkout clerk. The checkout clerks are wearing gloves. Hey, that's great, except for the fact that those gloves are contaminated the instant they touch a customer's product the very first time. After 4 hours of touching 10,000 products, those gloves are there is a very high chance with the number of people that they have come in contact with coronavirus on gloves that need to be changed between every person at least. And so the whole thing, the woman, to me, that was actually hysterically funny to me. It was actually funny that she has the N95 mask that is the one that healthcare professionals have shortages of, and they need those to be protected, and they use them properly with their training. And this woman has the N95 mask and actually turns it into a coronavirus trap. Right. Awesome. Love it.
What we did is we were trying to do it sterilely.
Yes.
We weren't using PPE.
We were not using PPE. We were just using common basic hygiene, preventing transmitting the virus by picking things up and putting them down. We dedicated to purchases. We decided what we wanted and we picked only that. We tried to pick it up in areas where we didn't think people would be likely to touch it, where the stalkers would be less likely. Maybe picking up from the bottom, picking it up from an awkward angle.
Well, you were the sterile person. You sanitized the cart and the handrail on the cart, and so I touched everything else.
Exactly. Really common technique with surgical technique when you work in pairs. One person is sterile, and they maintain that sterility. The other person is kind of the dirty person.
I was dirty.
And she was dirty. And they are the ones that can go around opening doors for the sterile person and things. So with two people, it can be a little bit easier. But how many people just don't do it right?
But it was all for nothing because the checkout person touched every single thing.
Exactly. The checkout person wearing gloves so she feels safe. Unwittingly, those gloves, everything she touched defeated all of our purpose.
And all the face touches as well.
Oh yeah, lots of people doing that. So unless everybody uses sterile technique in public,
it's not going to work for anyone.
Yeah, that's a problem. But if you properly use sterile technique, properly use your PPE, yes, it will protect you. Okay. I have a question for you.
Okay.
All right. Have to wear my glasses. Okay, here is a question that somebody asked, a very typical one that I get. You are a total clown. It is obvious, given the lethality of this virus, that it is a lab-created hybrid with HIV, the AIDS virus. Open your eyes, people. The government is a police state, and if this isn't evidence that you are under lockdown, under a police state, I don't know what else to tell you. All right. Now, I'm going to let Vicky, a non-scientist, answer that. He is saying that this is a lab-created hybrid with HIV, and it is intended to cull the population.
Okay.
Right.
So that's impossible because we would see it, and not only we would see it, everyone who's researching it would see it.
Yes. Right.
You can't hide it.
And what would we see in that sequence? We wouldn't just see bat coronavirus. According to what he's saying, what other sequence, what virus sequence would
HIV.
You would see HIV sequence. And at the introduction, I just showed you guys 100% match, well, 99.98%, no less than 96% with bat coronavirus. There is no HIV sequence.
Right.
Where is the evidence? Where is this guy's evidence showing a homology with HIV? Right. Okay, great. I'm going to ask you one more question and then it's all yours.
Okay.
All right. There are glasses. Okay. This is one all of you guys see in social media, some variant of this. There is no possible way this virus came here as late as December. It was way sooner than that because I am 100% positive I had coronavirus last October. I was sicker than I have ever been in my life, and yes, I took a flu vaccine. They are not telling us the truth about this disease. All right. Everybody who's watching this has seen, as I mentioned in video 1, everybody who is sick thinks they have coronavirus. As a scientist, what do you say about that?
Well, I'm not the scientist, so...
Okay, so let me rephrase that. As a scientific thinker, the woman says she is 100% positive she had coronavirus last October. She took a flu vaccine, and she was sicker than she ever was before, and she had all the symptoms.
Okay. So I believe in order to be 100% sure, you need to have taken one of the tests, and it has to have shown that it was positive.
Yep.
And it could have been any other strain of flu that we get every year. I mean, there are many different kinds, and they all have similar symptoms. So you really can't be sure that's what it was.
Exactly. You cannot diagnose it off the symptoms. You cannot do that. And the other thing is, I don't remember if we covered it in the first video, but the flu virus actually, there's no flu virus. There's many flu viruses, as you correctly stated. The vaccine only protects against 3 of them, actually. And even then, a flu vaccine is only about 43% effective at best. So every reason to think that it could have been anything else. All right, let's go on to the next one.
Here's one from Andrea and Charles. Is it true that ACE receptors are more prevalent in smokers than non-smokers?
These are the kind of questions that I love. So this is referring to, in the first video, that the virus binds to cells that produce angiotensin converting enzyme. And by the way, I think maybe you'll have pulled it up. There was a comment that it's actually ACE2, and that is accurate. But again, we're simplifying. The answer to this question, the intent of this question is because the virus binds to cells that produce angiotensin converting enzyme, or ACE, if smokers have increased levels of ACE, they would be more at risk. And the answer to that, I know because I spent only 18 years in pulmonary disease research. Absolutely. There is no question about it. I myself have produced data that clearly shows that there is increased levels of ACE expression in smokers. Smokers are at higher risk for getting the disease, and also most smokers have underlying conditions as well that would make them much more susceptible to the pneumonia that I described in the first video.
Okay. So they go on to ask, since you live in Colorado, is there a difference in ACE receptor counts between marijuana smokers and cigarette smokers?
As if marijuana smokers are only in Colorado. I don't know. I've never seen any comparison with marijuana. Any type of irritating inhalant to the lungs that elicits a inflammatory response, it's going to increase ACE levels, as well as interleukin levels, tumor necrosis factor levels, transforming growth factor beta, all of these cytokines that people mention. As a matter of fact, one viewer earlier mentioned when I was talking about the inflammation in lungs, he referred to it as a cytokine storm. Really good description. Anything that irritates or inflames your lungs is going to cause the expression of cytokines, which makes you more susceptible.
Here's one from Martin. Do you think there could be a link between ACE inhibitor high blood pressure medication and severe COVID-19 reactions?
Okay. So actually that was That was one of the things I was going to allude to in the last video. Now that you guys have this kind of scientific training and scientific mindset, you can start putting things together. I alluded in the last video that some people are familiar with angiotensin converting enzyme because they are on ACE inhibitors for things like high blood pressure, heart problems. So think about it. If ACE is required for the virus to bind to the cell, what if you were on an ACE inhibitor? Would that make things worse, or would it actually reduce the chance you could get infection? Does it make sense that if you have less ACE, less binding sites for the virus? So the answer to that is right now there is no information either way. Obviously, one of the experiments I would love to see is looking at maybe older subjects, some of whom are on angiotensin converting enzyme inhibitor, ACE inhibitor, and then equal age groups where they are not on it, and see if you can find that maybe one group or the other succumbs worse or better to the virus. No such data exists at that point. It is definitely worth experimentation.
Okay.
All right.
All right, next is from Gearstill. Why do you say that the viruses are not alive? I learned in biology that the life on Earth is represented by plants, DNA specialized in building what we call plant, animals, DNA specialized in building animal organism, and free genetic material that is called virus. In order for the virus to reproduce, it needs factories, so it steals the factory from the ones that build it. So is that not alive?
That's a good question. So no, viruses are not alive. In science, in philosophy, there's lots of different definitions of life and living. In science, there actually are two, but they all have a commonality, and that is there are at least 5 requirements that something is alive. And I actually don't remember what they are. One of them is it has to have a metabolism. It has to consume energy, use energy. It has to be able to basically use food, so to speak, and viruses do not do that. The other one is it has to be responsive to the environment. Viruses do not respond to the environment. It doesn't have to be a conscious thing, like, wow, it's hot in here, I'm going to move or react. There are other things where it's strictly mechanical or chemical, things like chemotaxis. There are some organisms that, moths, for example, they just go to light, not because they like it, but they just do because it's a mechanical process that they're wired to do. Response to environment. Reproducibility is another one, and viruses do reproduce. Another one is energy.
Oh, no, that's
reproduces. Well, all right. Well, actually, I tried to think of what the other ones are. I can't. The bottom line, though, is a living thing has to have all of them. If you miss even one, it's not living, it's not alive. And viruses have 2 of them there that we were able to find that they don't do. So, yeah, I can't remember what the other ones were.
That's all right.
All right.
You remember a lot, so.
I try.
All right, so from V-Way, ELISAs are not necessarily for detecting antibodies, though. They can also be designed to detect antigens, in this case indicating the presence of Sars Cov2 itself.
Damn this guy. All right, yes, he is correct. All right. All right, now we're going to need to use the board. I figured we probably would at some point. All right, this guy's obviously a scientist, very good technical question, and I want to be accurate. In the previous video, I had stated, correctly, by the way, there are 2 tests that are generally being used. One of them is, I think you guys remember, the qPCR test, the RT-qPCR test, which tests for the, ultimately, you're looking at the RNA in the virus being in your cells, the viral RNA being present in your cells, clear indication you have the virus in your body, you are shedding it. The second one was the ELISA test, which I said looks for antibodies. And the antibodies would tell you you could either have it currently or you had it before at some point, but of course you still have those antibodies. The other way that you can do it is you can do the ELISA test backwards, so to speak. So let me show you just this is going to be a huge oversimplification of an ELISA test. Okay, so the way the ELISA works, as I described, is you've got this little sample dish. And this little sample dish, by the way, is like the size of your pinky fingernail. So really, really, really small. And in the sample dish, what you have is you will have bound to the sample dish the antigen. You will basically just have the region of SARS-CoV-2 that the antibody recognizes. And actually, you know what? In the previous video, I did triangles, didn't I? Yeah. Yes, so we're going to do triangles. So you have your antigen, the thing that the antibody would recognize. So you have these bound in the bottom of your plate. You then add the patient's sample, and generally with this test you would use blood. This wouldn't be quite like the swab test. You wouldn't do the swab test on this. But anyway, you add that typically blood sample, and then within the patient's blood there's going to be the antibodies as well as a lot of other things in there, lots of stuff in blood, as you can imagine. But one of the things is if they have the antibody for SARS-2, well, then those antibodies are going to bind to those antigens. And then when this happens, and there's different ways of detecting it, but when this happens, there's going to be some type of a reaction that is detected, either colorimetric, light, something. But when this interaction with the antibody and the antigen occurs, it gives off a signal. When that signal is given off, we know there were antibodies for SARS-CoV-2 in the sample this patient either has or had it. Now, what this gentleman's saying, and he is very correct, is that you can do it backwards. What you do in this case is you start off in your ELISA with the antibodies stuck to the plate, not the antigen, not the part the antibody recognizes. Pretty smart viewer. So you have the antibodies bound. Same thing. Add your patient sample. Now, your patient sample also, of course, has antibodies in it, but it also, if the person is currently sick with SARS-CoV-2 and expressing the virus, shedding the virus, there will be antigens in that sample as well. Some of the antigens can be bound to the antibodies, as a matter of fact, but there will be antigens in there. And those antigens fit. Either way, you have an antigen-antibody reaction. There is going to be a signal that is given off. We can determine that this patient has antigen for SARS-CoV-2. A nice thing about that is that you would know that patient is currently shedding virus. So it is possible to do that. The reason I gave the other example is because when I looked at how the test works, it absolutely, the instructions that I read showed that it is an antibody test. So still accurate. You could do it this way. This way also, there's reasons to believe it would be less sensitive. ELISAs are already not extremely sensitive assays, but you could do this. Wow, that was a doozy of a question. I got to be on my toes. All right, did that make sense?
It did.
Okay,
yes,
good.
That was a good question.
That was a damn good question.
Yeah. Steve would like to know how it's possible for someone to have tested positive to CV but have no symptoms that can spread the virus. In other words, how can the virus replicate unmitigated by antibodies to the point where one is contagious but the body never reacts?
The reason is because the virus is still transmissible from those people because it is still... First of all, they may not be sick yet, all right? Some people, it just takes them longer. Remember, the immune system, the person's immune system is still fighting the virus, all right? And for some people, they'll get sick very, very, very quickly. Other people may take many, many days before they get sick. Others won't get sick at all. But either way, they're still shedding the virus within their bodies. Now, they may not cough and produce as much as someone who's full-blown sick, but they still are shedding the virus even though they're not sick yet because they just don't have enough levels where they're feeling the impact yet. Remember, it only takes one. You just need one virus infecting the cell, and it starts copying that RNA, and now you've got hundreds of them, And then when the cell dies and releases those, now you've got thousands of them. For some people, after that first cell dies and the other viruses go out, their immune system just happens to pick up most of those viruses. Huh. But there are some that still get away, and they just take a longer time, so they may not get sick.
Okay.
Yeah. Most people get some type of symptom, though. Most people get some type. Yeah.
At least one, huh?
Yeah, and again, you may not be coughing and sneezing, but it's still in your bodily fluids. You can still transmit it through coughing, sneezing, surfaces, things like that.
Okay.
Yep.
All right, next we have Lamp Man. He wants to know, if the virus is transmitted through the air, how did the first human get it? Or was this created? Well, that goes back to our first discussion about created.
Yeah. Well, let's. Well, first of all, let's see if you can answer. Is it possible that this virus was created in the lab? Because again, a lot of people claiming that this is a government conspiracy thing and stuff. Not that this guy is. This guy isn't. But could this be created in the lab? Is it possible?
Well,
do you think?
I think if you have the unique signatures, it's possible.
Meaning?
Meaning, yes.
Yes.
Yes, I think it could be created.
All right, so the answer is yes.
But I would think it would take quite a lot to do it.
Actually, no. No. In a laboratory, we can create synthetic DNA. We can create synthetic RNA. You can create, because in the laboratory, we can just take the A, C, Gs, and Ts. Mm-hmm. We can use an RNA or a DNA polymerase, and we can create a DNA. We also have machines that these machines will produce DNA and RNA and big pieces of it. You absolutely could produce this in a lab. But from a bioterrorism perspective, can you think of a reason? First of all, we know the sequence is already back coronavirus, so it's already done. You don't need to create it in a lab. It's already there. The second thing is, if you were to create something like this in the lab, can you think of reasons why we would say that this one particularly wasn't created as a weapon of mass destruction or something?
Well, if you're going to create a weapon of mass destruction and you have that ability, you would think you'd make it stronger, make it move faster, if you're going to create, go big.
Yeah, exactly. I mean, if this was a lab-created weapon of mass destruction, it was created by Dr. Evil from the Austin Power movie, okay? Why would you create something with the R naught of only 2 that has less than a 1.5% mortality rate? When we talked about measles, why wouldn't you mutate measles to an unrecognizable form? R naught of 12 to 18 and far more virulent and deadly. It's silly. It's just silly. Yeah.
Okay. So what about his question about it being transmitted through the air? Then if that's true, then how did the first human get it?
Yeah. So not entirely sure on that. And also do keep in mind that with my particular skill set and background, I'm the kind of person that would come in after the fact, so I'm not really an expert on the transmission across from animals to humans. There's various ways that could have happened. I think the people who are experts on this do believe that it wasn't a direct transmission from a bat to a human. They believe that there was, what is it called, a penguin? There's another animal that bats kind of seem to have an affinity for hanging around or something like that. And the people in China eat these penguins, and it's believed that maybe the bat contaminated a penguin, and then if you eat something, well, it's pretty damn close to your airways if you eat something, right? And that a human may have had it transmitted. Those are things that I actually don't know, so I don't want to answer them too much. But from what I've read and when I read literature, I don't get it off Yahoo News, by the way. I get it off of different sources, like PubMed is my favorite source. It is a public access that you can get actual medical research. Actually, I've got publications on there. The only problem with PubMed is that you really have to be a scientist to understand the stuff. It reads really highly technical. It's not something most lay people can read and go, oh, I get it. Yeah, I wish I could answer that question a little bit better, but I can't give you a definitive one. Yep.
All right. So Lampman also wants to know, can this spread through mosquitoes?
Can this spread through mosquitoes? Do you want to try a crack at that one? That's a good question.
Not.
You do not? Okay. Okay, so this is one where, again, you want to fall back on your scientific knowledge and understanding the mechanisms with the virus. All I can say about that is I cannot see any way that that would happen. And I don't want to go into too much other detail, but which I know defeats the purpose of the video. We're supposed to be talking about things that you guys could answer through our training, and it would make sense. I don't see that happening. There has never been a case of transmission that has been shown to be from mosquitoes. There are people that they basically do things like that, though. They look at mosquitoes, they see if mosquitoes are carrying Cov2. That would be reason for concern right there, right? If you never find any mosquitoes that are carrying Cov2, well, then there's a lot less concern. The idea of it being transmitted by mosquito, I cannot say that it can or cannot. I am saying that it is extremely unlikely. Extremely unlikely.
Okay.
Yep.
Right.
Good question.
Okay, so from Nicholas, this has been, I've seen this all over Facebook. I've seen it everywhere in some form. It has been advised not to take ibuprofen because the virus is now thought to feed off of it. Is this true?
Oh, you know what? That's one you can answer.
Okay.
Yeah.
Well, my first instinct is no, because as we discussed, it's not alive.
Right?
I'm sure you have better reasons for it.
Just the fact that the virus isn't alive, that actually isn't enough. Remember, there were 5 things of life. What is one of the criteria for life that the virus lacks that would dismiss that as being untrue?
Metabolism, was that?
Metabolism, right.
That was one of them?
Yeah.
I'm sorry, I didn't retain the rest.
That's all right. That's all right. Because you're correct. Okay. You're correct. Metabolism means that you are expressing energy. In order to express energy, you have to have food.
Okay.
Viruses do not have metabolisms. They do not express energy. They do not consume energy. They are just things that stick there. So they do not eat, and therefore ibuprofen is not food for them. They don't eat off the ibuprofen. They don't eat off anything. They don't even eat off the cells. They just simply reproduce within them, but they don't eat or consume anything.
Okay. Shrapik wants to know, how long does it take to do the RT-qPCR test in a lab? What is the main limitation here, and how many PCR machines does one lab usually have?
Okay, great question. This is right up my alley. It's what I've done all the time. So when I do a RT-qPCR, I typically can complete a full experiment from sample preparation to looking at those curves within 3 hours. The limitations of it really are the number of samples. If I'm doing samples for an experiment, maybe do 15, 20, sometimes as many as 96. Most of the people that are going to be doing this testing stuff, they'll be doing as many as 384 or more samples. It takes time to do that, to set up those samples. So the rate limiting factor is setting up the samples. It takes longer to do that than it does to run the machine. The sample setup and everything takes half an hour for, maybe if you're doing a reasonable number of samples. Probably takes you most of the day if you're going to be doing a whole bunch of them. The PCR machine itself only takes about 2 hours before you can get the data. There was another part to the question, and that was...
Let's see, how many PCR machines does one lab usually have?
Yeah, so for a typical academic research lab where I came out of, you would basically have one machine for a department a lot of times, so as many as 5 or 6 labs, maybe even more. Very seldom that each lab has their own. At other companies I've worked with and everything, I have seen companies that do this type of work that have had dozens of them, a whole room full of these $60,000 PCR machines. Uh-huh. Yep. But again, the rate limiting factor is you still have to have people prepping the samples. Okay. That's why it takes like 3 days or so. It doesn't really take 3 days to run the process. It's 3 days because it just you gotta collect the sample from a patient. You gotta mail it in or bring it in. People have to get the PPE on and everything, prep the samples. So that's why it's a few days.
So the next question is from Dreadnought. He says he'd like to point out that it's not ACE, but specifically ACE2, that the virus attaches to.
Yeah, we covered that one already. And yes, that is correct. It's specifically, there's different isoforms of ACE and technically ACE2. That is correct. I just didn't want to overcomplicate things, but he's right.
There's just a whole lot of stuff in there.
Yeah. Okay, yeah, so this guy's actually got a lot of really other very, very similar technical questions. One of them he's saying is that the coronavirus matches that he has seen the closest is over 90%. We covered that at the beginning of the video. Also that the RNA genome is bigger than 24 kilobases. Yes, I covered that. He says it's more like 30 to 32 kilobases, 30,000 to 32,000 bases. As I showed you, it's just about 30,000. Other stuff on coronavirus. Yeah, no, the guy's pretty well informed. Yeah, I mean, we're going to have some scientists that come along and see this, but again, I'm trying to produce this more for lay people, so I oversimplify.
Okay. Let's move on. So here's a question from a subscriber. How do macrophages see the virus with the antibody attached, and how do they know it's even there to attack it? Is it emanating some kind of chemical trail for them to follow?
Oh, really good question. Yeah. So the answer is no, it doesn't do chemotaxis, which would be some kind of chemical signal or trail. Basically what it is you've got your virus. I think you know about my drawing skills. And then you've got your antibody attached to it. And then the question is, you've got this white blood cell. How does the white blood cell know to eat that? And the white blood cells do eat that. White blood cells are alive. They reproduce, they express energy, they react to environments, things like that. So basically, it's not that it goes looking for this. It doesn't have a line of sight to this. It's just that they're kind of going around doing their thing. But when they come in proximity to this, they have receptors on here that basically, this antibody kind of looks more like that. And then that sticks onto the receptor. And it's kind of like using a magnet to just kind of comb along the carpet. And if you happen to find something magnetic, it picks it up. So it's more like that. It's not that it actually actively looks. That's a pretty good question.
Okay. All right. Okay, so Jim wants to know, do we necessarily need a doctor to see the patient or suspect patient? Since they probably don't do the testing themselves, why couldn't a med tech or a nurse do it?
You know, I don't know. I'm not a doctor. I can't really answer that. I can't. Yeah.
I'll skip it. So here's one from Mark. He is... He's asking questions because he's missing his pub buddies, and they discuss these things. They have these big questions.
Probably people discussing this at a pub is where we get the misinformation, I'm guessing.
Yeah, he has a few questions.
All right.
He starts out with Russia, Iraq, and China have seen comparably very low rates of infection, and Sars Cov2 appears to be the perfect biological weapon. We talked about that.
Yeah.
Is there any possibility that it could have been engineered? But we discussed that.
We discussed the bioterrorism aspect. It is not. Also, remember, we have. Why isn't that claim made about every disease and infection we have? However, you can actually, given our epidemiology, you can answer the first one probably because you're allowed to give an opinion. One of his things is China is reporting really low new infection cases, really low. What's your thoughts on that, given the biology of the virus and what we know about epidemiology? Any thoughts on that?
Well, my immediate thought is that China's not being completely honest about their reporting.
Okay, now, when we say something like that, we've got to give evidence. We have to get, not only evidence, we have to give logical, rational, scientific reasons that would be true. Otherwise, we're no different than the people on the internet. So what would be a logical, scientific reason that they're not reporting accurately? Okay.
I don't know how you can go from blowing up to having absolutely no new cases.
Okay, so you're saying...
So what did they do?
You're saying that they basically went here. I forgot how many cases in China. I don't remember how many in China, but let's just say that it's 500,000. Well, let's see. Actually, U.S. That's worldwide, so it's less than 100,000 because I remember reading recently that the U.S. has surpassed all else in the world with the number of coronavirus cases at whatever 100,000. So let's just say it's 100,000 cases. 100,000 cases, and then it leveled off. No new cases after that. All right, what are some challenges that you have with that being what they're actually reporting?
Well, their population.
Population.
Count.
Okay. What's the population of the United States? That's okay. The population of the United States, where by the way, this is the U.S. Okay, we're still passing it up. U.S. population right now, it's right about 340 million.
Okay, so I would have gotten there. Okay.
Okay. Do you know the population of China?
I do not.
Okay, it's 1.5 billion.
Okay.
Billion. That's like almost 5 times the population, 4.5 times maybe population of the United States. So we got a lot more population, a lot of people closer together, and still they have less cases and no new ones. Okay, we need one other factor for this that really makes this maybe a little suspicious. What's that?
Why don't you throw it out there? Oh, okay.
R naught.
Right.
What was the R naught?
Yeah, well, it was at 1.2.4.
Okay.
Right.
All right. So the problem with this, and I'm trying not to influence here a little bit, but you guys can do the math. China's on this up curve, and then all of a sudden they got no more new cases, despite the fact that everybody is around each other, and every infected person on average affects 2.4 others, but it stopped. We know it isn't because everybody in the whole population got it, because there's not 1.5 billion cases, right? How do you explain that? How would you explain that given the population, the R naught, and another country with 1/5 the population still increasing exponentially?
That's a question for China.
Yeah, I think it is.
Yeah.
Yep. Or for these guys.
Okay. I'll let you guys answer.
Yeah, you guys tell me. I can't explain it, but my job isn't to judge here. We're just giving information. Those are your statistics. That's your epidemiology. You come to the conclusion.
Okay. So Mark actually had another question that I thought was interesting. He's in Australia.
Oh.
And he's saying, as I'm sure many places do, we've had issues with bats spreading disease through their droppings, their urine, and saliva. Assuming that Sars Cov2 is endemic to only the bats that have been exposed to the virus, could we as humans pass the virus back onto the local bat population? Wait. Yeah. And if bats do carry virus, would it be transmissible to humans through their excretions and saliva?
He's referring to what I believe is called zoonotic transmission. Zoonotic transmission means across animal species.
Okay.
To be honest with you, I am not really well versed in that. That is a fantastic question. Maybe somebody can answer it. Again, we know the criteria for answering it. You don't just answer it. You give sources. You make it so that somebody with the education that we have can say, that makes sense. But you don't just answer that. I don't know. I can't really answer that. There are cases where people have transmitted viruses to like their pets and vice versa. It's well established that people with birds very often can get virus from birds. There's lots of avian viruses that are transmissible. For the coronavirus, I definitely can't say anything though.
Well, here's one more. It's probably not for you, but maybe someone out there. It says, under the right conditions, could the virus survive and reproduce outside the animal host, say in rainwater or in an improperly sanitized pool? For instance, a bat drowns in a water tank, already full of organic matter, phosphates and nitrates, and sitting at 36 degrees during summer.
I don't know.
It's the pub.
I don't know where that guy. Yeah, I guess it's the pub.
I think he's awesome.
Won't be going to a pub in Australia anytime soon. All right. Well, it's you know what? That's a question you might be able to answer. You might be able to answer.
So.
Yeah, I think so. You might be able to answer that one. So let's simplify it again. The question is, you have a bat that has the virus, and the bat dies, and he drops into a tank of water that's all yucky and gunky and everything like that. Can the virus replicate in that bat, and is that a contagious scenario? I think you probably could answer that.
I don't think I should.
Why not?
I don't want to mislead anybody. Go ahead.
No, you're not going to mislead anybody because I'm going to correct you.
Just do it. You've got it.
I think you got it.
No.
Nope.
No.
Okay. All right.
Put my foot down.
Okay. That's how we argue, by the way. So the answer is yes, that could be potentially a problematic situation. First of all, until the bat is completely dead, every cell in the bat is dead, that virus can continue to replicate and get to other cells. Now, it's going to be very difficult for it to do it rapidly because, again, there's no bloodstream and things like that. The cells are going to die rapidly. The viruses won't be able to travel as quickly because the bat's it doesn't have any circulation, right? But those cells in immediate proximity that are still alive, yeah, their functions, as long as they're alive, they can produce virus. So now you've got this dead bat that's got a decent amount of viruses in it, that as the bat decays, those viruses can be released into that water. And if that water has these conditions that the guy is basically saying can support not degrading the virus, yes, somebody who came by and drank the water or splashed it around or something like that, yeah, sure, you could get it transmitted that way. Maybe an animal comes by, and gets the virus, and then you eat the animal, something like that. Which, to be honest with you, eating the animal, if you ate the lungs raw and then you breathe it in, something like that, I mean, I don't know. I don't know what they eat in Australia. I don't know. I thought they'd just drink that Foster's beer. An Outback Steakhouse, that's all I know.
Okay, so I'm really glad that you answered that question.
It's theoretically possible, but yes, it is theoretically possible.
It's a good thing you did that one.
Okay.
Because that's not where I would have gone.
All right.
Okay, so next from Caduceus, how about alkalizing our body with better diet and perhaps some sodium bicarbonate? I read that sodium bicarbonate was effective during the 1918 flu.
Yeah, okay, so you guys have probably seen, where did you get that question? Wow.
Off of
I don't remember. Okay. So the answer is, I cautioned you guys about avoiding any of the homeopathic stuff and everything. That stuff works great when nothing's wrong. When something's wrong, you need to stick with things that you're limiting your variables because some of these things can hurt you. You've seen the alkaline water. You ever see the alkaline water and it advertises, I want to say it's pH 8.4, maybe 8.0, 8.4, something like that. And the idea is that when you drink the alkaline water, your blood pH is 7.4. When you drink the alkaline water, it brings up the pH. Do we know what pH is, by the way, when I say pH? Are we aware? I don't know. I'm asking. You're them. That's why you're here.
I need to know about it. Go ahead.
Okay. Well, okay. Do you know what pH is?
Not really.
Not really? Okay. Do you know what acids and bases are? Okay. So basically, pH 7 is balanced. Your blood pH is like pH 7.4, but that's balanced. Anything lower than that is an acid, and anything higher is a base. Okay, so blood is just very, 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 very slightly basic, very slightly. The idea is that if you drink this high pH base, that you will increase the pH in your body, which can destroy viruses and everything, because it actually is a fact that if you have the virus and you put it in something pH 8.4, that virus is going to denature. It's going to kill the virus. Also, if you put it in an acid. The problem with that is that your blood is what we call a buffer. And you can drink all the pH 8.4 water you want. It is not going to change the pH of your blood. As a matter of fact, it's exactly the opposite. If you were even able to take one drop of your blood and put it in a cup of that water, it would bring that water to pH 7.4. Okay? So you can't change the pH in your body. So don't even bother with that. Yep, total myth.
Okay.
Yeah, if you could change the pH in your blood, if your blood pH went to 7.6, you would die.
So here's one that I think we've already covered, but why not exchange social distancing to an obligation of wearing PPE, like Japanese and Korean societies have done? It will limit spreading greatly from an R naught to less than 1.
Yeah. This is one you can answer.
People cannot be relied upon to use the PPE correctly.
Yep, exactly. Again, wearing the PPE is only effective if you use proper sterile technique, and the overwhelming vast majority of people in the world by far have no clue how to use the PPE. So, yep, that's where the problem is. Absolutely.
Oh, here's a good one. So, Turbo Buick.
Turbo Buick, back to car stuff.
That's one of your car guys.
Yeah, that's got to be a car guy.
If you get the coronavirus and recover, will there be any permanent damage from having had the virus?
So there can be. Do you think you can answer that one?
No, I think you should.
Okay, that's all right. That's all right. The answer is it can happen. You will hear reports that if you get the coronavirus, you will get permanent lung damage. That is absolutely a false statement. That is just false. If you get pneumonia, you may get permanent lung damage. And I think that's the best way to answer it.
Okay.
It's not just with coronavirus. Any pneumonia can result, because remember I explained very clearly how this works, and you get the destruction of lung tissue and cells. When that grows back, there is the possibility you could get scar tissue, and that it could be permanent. So it's true for pneumonia, and the answer is yes, it could be permanent. Okay,
so one more, and this is what prompted you to think, wow, I might do a video.
Okay.
Because we were looking through Facebook, with my anxiety growing,
and
I'm reading you all of these different posts, and I'm saying, is this true? Is this true? Is this true? And I know I drove you crazy.
No.
Here's this one that has been circulating, and credit is given to a well-known hospital. I'm not going to say which one, because I don't think it actually came from there. Maybe it did, but got, the game of telephone, it got changed and changed and changed and changed.
Okay.
But the very first statement on it,
all right.
This virus is not a living organism, which we know is true, right?
Right.
But it's a protein molecule, parentheses, DNA, covered by a protective layer of lipid fat, which when absorbed by the cells of the eyes, nose, and mouth, changes their genetic code, mutation, and converts them into aggressive and multiplier cells.
So that is a statement by a medical doctor.
On Facebook.
Okay.
It's a statement by
somebody on
who
Facebook.
knew somebody who had a doctor who had a cousin who was a doctor.
Probably.
I don't know.
Okay. Well, here's the bottom line. You can answer that. You can answer that. So first, before we do that, let's draw it out. Okay. So I'm going to draw it out as you read it. Okay.
Give you some practice on that.
Okay. So non-living. Okay.
It's a protein molecule DNA, whatever that means.
Okay, and can you read that again? A protein? Can I read that?
Sure.
Is that word for word as it was?
That's how I pulled it off of the million.
Okay. It is a So it is a protein molecule.
Parentheses DNA.
Parentheses DNA. Okay. So that makes no sense.
Okay.
But so it has DNA, and basically what it's saying is that the whole thing, the way that reads, is this is a protein made of DNA. So the way that reads is that this whole thing is made of DNA. The whole protein is made of DNA. Okay, let's go.
Covered by a protective layer of lipid.
Okay.
Parentheses fat.
So it's got some fat. Yep, lipid is fat. Okay.
Which, when absorbed by, and here I've paraphrased, cells of the eyes, nose, and mouth.
Okay, so now it gets into what we call a somatocyte. Well, not a somatocyte. It's going to be an epithelial airway cell because it's Sars Cov2. So it gets into an airway cell.
Okay.
And okay, so now we have our protein DNA in the cell. Okay, now what happens?
It changes their genetic code.
Changes their genetic code. Okay, so cells have a nucleus where, again, your own cells have your own DNA. They have your human DNA. So we've got our DNA, all of our chromosomes in this nucleus. So what it does is it goes into the nucleus and it changes your DNA. Okay, keep going.
Then there's, in parentheses, mutation.
Causes mutations in your DNA. Okay.
And converts them into aggressor and multiplier cells.
And then converts the cell to attack other cells,
Okay.
is what that reads.
Okay.
So let's start off here. This is a protein made of DNA, is what it says. Any comment on that?
It's wrong.
That's wrong.
It's RNA.
It's RNA.
There is no
Good for
DNA.
you. There is no DNA. Second of all, are proteins made of DNA?
No.
No. Proteins and RNA and DNA are completely separate
Oh.
things.
Okay.
Okay? So, exactly. So that's wrong. It's covered with a lipid layer, a fat
That's
layer.
good.
That's true. Got that one? That's right. It's non-living.
Right.
That's right. Hopefully you guys are following along too. I want to make sure...
If I can follow this, anybody can follow.
These guys should be following along with this if they watch the first video. It's a protein molecule, DNA. DNA is a protein molecule.
So it's wrong because we're
That's
referring
wrong.
back to the beginning.
Yep,
It has
that's
nothing to do
wrong.
with DNA.
So far they got two things right. It gets into a airway cell. I think it said that. Did it say it gets into...
Yeah, when absorbed by the cells of the eyes, nose, and mouth.
Okay,
Yeah.
got that one right.
Well, that's good.
Good. And then what happens is it goes and it changes, it mutates your DNA in the cell.
It can't because it's RNA, right?
So first of all, there are some viruses that can do that. It would have to go in there and change the DNA, mutate the DNA. Does it do that? Do you remember how the virus worked last time in the previous video?
Not sure.
Okay. The RNA from the virus gets out into the cell and makes copies, and then ribosomes produce more virus. Okay? Does that equate to what this person is saying, that the virus causes your own DNA to mutate?
No.
No.
It just multiplies and...
Yeah, it multiplies. Your DNA is in the nucleus of the cell. None of this is happening. None of the viral replication for Sars Cov2 happens in the nucleus.
Right.
None.
It's multiplying all around it.
Exactly.
And making
Yeah.
the person sick.
Multiplying all around the nucleus, but it does not change the DNA.
Okay.
Right. It just simply hijacks the organ called the endoplasmic reticulum.
So glad that you threw that out there.
Then what it does is it reprograms the cell to become, what is it? A killer.
No. It changes the genetic code.
Changes the genetic code of the
Converts
cell.
them into aggressor and multiplier cells.
Aggressive. So what it does is it now programs this cell to replicate and kill other cells. Correct?
That's what they're saying.
Is that correct?
No.
No, that is absolutely ridiculous. Okay, even the AIDS virus doesn't do that. It doesn't program your cells to suddenly kill other cells. That's ludicrous.
So that is why I brought it up, because
Yep.
there's so much misinformation out there.
There is a lot
And
of
if you
misinformation.
attach the right name or the right hospital, or say a friend of a friend of a friend's doctor said this, it suddenly becomes credible.
Yep, and
It's
also
scary.
it sounded medical.
It did sound
It sounded medical.
But it's scary,
Yeah.
and it's increasing
Yeah.
anxiety.
And only
'Cause that
two
sounds
things are right about it.
horrible.
Sounds pretty bad.
Yeah.
Yep.
Okay.
And I think most of you guys could also have followed along there. That's an interesting resource. By the way, just to let you know, I believe I have seen that resource, and it is supposedly from a doctor at a very, very, very credible institution. But again, we're going to default to the probability that it was reworded by a layperson, maybe.
Right.
Yep. I hope.
We're hoping.
I hope. All right. All right. Well, guys, that's pretty much it. If you've got other questions, we may do another one of these follow-up videos, and maybe if something big and earth-shattering comes out or whatever, we can update on that. But again, we just want you guys to be informed and help fight against the misinformation, which unfortunately I think is still winning.
And
Although we're doing pretty good.
your anxiety, too.
It does lessen anxiety. Would you say your anxiety is lessened?
Oh, massive
Good.
amounts, yes.
All right, good. Hopefully for you guys too. Thanks for watching. Hope you found this helpful. We'll see you next time.